Hello there. This is number 5, a listed researcher of Horizontal 2020 FoodSmartphone project. In general, the FoodSmartphone project aims to develop smartphone analyzers for food safety and quality analysis. I joined the project in late 2017 as an enlisted researcher based at Queen's University, Belfast. My individual project aims to develop software tools to enable user-friendly and automated signal processing for biosensing and data transmission among stakeholders. During the flashing 3 years period of time, I have made a couple of progress towards the vision of the project, together with a bunch of incredible people, including my supervisors, colleagues, and friends. Here, I would like to walk you through some of the innovations that we have made during the course of my research and study in the project. The first piece of work that I would like to share is a smartphone-based modulated colorimetric reader, or namely SMCR. One of the major drawbacks of current smartphone analyzers is their limited ability to support various smartphone models and bioassays for food analysis. Potential end users, especially food consumers, hold smartphones with very different models, dimensions, and specificities. Up to date, the majority of smartphone analyzers in the literature were designed for a specific smartphone model and bioassay, which limited their applicability to be used in everyday scenario. The SMCR innovatively introduced the modulation concept into the design of smartphone analyzers. The SMCR modulated the reader into 3 parts: a smartphone case module to adapt various smartphone models, a light source module to support multiple types of light sources, and an assay platform module to enable coupling with numerous assay platforms such as little flow devices and microfluidics. A 3D printed SMCR prototype costs less than £6 in total, making it affordable even by people living in resource-limited regions. To demonstrate the applicability of the developed SMCR, we use pH testing of red wine as an example. Controlling pH is essential for winemaking. Currently, there are 2 ways to test pH of wine samples, either using a pH test strip or handheld electrochemical pH meter. However, when people buy pH test strips that are claimed to be designed for wine pH testing, they found that the pH test strip is unable to test pH value of red wine due to the red color interference. Lots of low review ratings were given to the product because of that. Even worse, if the volume of the red wine sample is not large enough to cover the sensing probe of an electrochemical pH meter, there will be no solution available to test pH of such wine sample. Together with the SMCR, we have proposed a novel color subtraction algorithm in the software pipeline to subtract the color interference due to the colored solution. The overall workflow of the software pipeline is shown in this diagram. Firstly, the smartphone camera response nonlinearity is calibrated through calibration. Then, testing images taken by the sample smartphone camera will be color corrected using calibrated nonlinear profile. As you can see in the second image, a solution color catcher is added to each conventional pH test strip to collect the color of the solution. The color of the solution will then be subtracted from the color patch or pH test strip. Eventually, pH of colored solution will be predicted by conducting the color regression on the subtracted color. From the experimental results, we see that the SMCR, together with the proposed color subtraction algorithm, achieved state-of-the-art pH predicting accuracy in terms of average error in pH value using pH test strips. It also demonstrated high reproducibility and applicability through experiments. Interestingly, we observed from the experimental results that lower-end smartphone models, on the contrary, produced a higher accuracy. We think the explanation is the more sophisticated image post-processing procedures embedded in higher-end smartphone models. Another work that I would like to introduce is the smartphone video processing app for the automated food biosensor interpretation. Currently, smartphones have been used to interpret little flow amino acids by taking photos of the performed amino acids at a set time and identifying the physical appearance of the control and test lines given the locations of those amino acids in the photos. A smartphone app that automatically interprets the little flow amino acids without the need of manual operations will greatly improve its accuracy and applicability by avoiding human error and promoting ease of use. In the project, we have developed a smartphone app for the multiplex allergen little flow amino acid interpretation. The app consists of two modules in the software pipeline: a lateral flow device detection module and a predicting module. The detection module was achieved using a state-of-the-art convolutional neural network model, while the predicting was implemented using an empirical model. The video demonstrates the interpretation process of the smartphone app. The lateral flow device detection module recognizes and locates the biosensor presented in the scene. Firstly, the video stream taken by a smartphone camera is decomposed into image sequence. Every image is fed into the detection module to recognize the region of interest on biosensor and its bounding box. Then the time intensity feature from the region of interest is provided to the empirical model, which calculates the final predicting result. Last but not least, a two-tier food analysis model for analytical data generation, interpretation, and storage, named Food Testing, has been proposed. Within the model, smart analyzers and lab equipment are utilized for distributed and accurate analytical data generation. Cloud computing is used to provide elastic computing resources, while edge computing on smartphones is efficient for data interpretation and is accessible even when internet is unavailable. A blockchain and database two-tier hybrid data storage scheme has also been proposed, combining the strengths of both. Blockchain is used to securely store food testing results and keep those stored data immutable and interoperable, while traditional databases are used to efficiently off-chain store large-sized analytical raw data. Together, the food testing provides trustable, efficient, and interoperable food analytical data generation, interpretation, and storage. At the end, we demonstrate a complete food testing workflow using the developed prototypes. Firstly, food testing result is generated using the SMCR. The food testing results and the biosensor image or videos taken by the smartphone are sent to the food testing cloud services, together with the geographical coordinates of the test performed. In the cloud, these data are processed and securely stored in a two-tier hybrid data storage. A MapView web page can be accessed to visualize all the trustable geographical food testing results stored in the food test chain. Thank you very much for taking your time on watching this video. I hope by further research and developing on smartphone analyzers and data transmission techniques, we can approach a safer and more authentic food supply system, and eventually promote public health.